Good morning, everyone. I'm Muhammad Nakyuddin. We're from Group 9. We present about biofungicides. Let's dive into the fascinating world of biofungicides, nature's answer to sustainable crop protection. So, what exactly are biofungicides? These are biologically derived agents that harness the power of natural organisms like bacteria, fungi, or plant extracts to combat fungal pathogen in crops. Think of them as the biological warriors that protect plants without the harmful effect of synthetic chemicals. Why are they revolutionary? Firstly, biofungicides are a cornerstone of IPM, an eco-friendly farming system designed to reduce chemical pesticide use. Unlike chemical fungicides, they specifically target harmful fungi while safeguarding beneficial microorganisms in the soil, enhancing overall biodiversity. They are also instrumental in controlling devastating fungal diseases in crops such as vegetables, fruits, and oil palms, ensuring not just healthier plants but also safer food for us. Beyond their effectiveness, biofungicides meet the growing global demand for sustainable and organic farming practices. By reducing chemical residues in the environment, they protect our ecosystem and contribute to long-term agricultural productivity. Imagine a world where crops thrive without harming the environment. That's the promise of biofungicides. First, environmentally friendly. Unlike chemical fungicides, biofungicides leave minimal chemical residues, making them safer for wildlife, environment, and earth. Think of them as the guardians of ecological balance, allowing farming to coexist with nature. Second, promote sustainable farming. With growing consumer demand for eco-friendly agriculture, biofungicides empower farmers to produce clean, sustainable crops while preserving soil health. They are the key to meeting market needs for organic and residue-free food. Third, pathogen-specific action. One of the standout features is their precision, where fungicides attack harmful fungi while leaving beneficial soil microbes and pollinators unharmed. This not only prevents unnecessary damage but enhances the natural biodiversity of farms. Fourth, resistant management. Pathogens adapt quickly to chemical fungicides, leading to resistance. Biofungicides, however, have diverse mechanisms of action, slowing down resistant development. It's like staying ahead of the game in the evolutionary arms race between crops and pathogens. Now, let's uncover the science behind how biofungicides protect crops. First, direct antagonism. Imagine a fungal pathogen trying to invade a plant's roots, enter biofungicides like Trichoderma spp, which release enzymes such as chitinase. These enzymes shred the fungal cells walls like scissors, stopping the invasion in its tracks. Second, competitive exclusion. Here, biofungicides play the role of bouncer. Beneficial microbes crowd out pathogens by outcompeting them for nutrients and space, making it impossible for the harmful fungi to establish themselves. Third, induction of systemic resistance. Some biofungicides act like a vaccine for plants. They prime the plant's immune system, preparing it to fend off fungal attacks more effectively. This triggers a chain reaction of defenses, fortifying the plants against infection. Fourth, biochemical interference. Finally, biofungicides can disrupt the growth and reproduction of fungi by releasing toxic metabolites. These compounds interfere with the fungi ability to spread, effectively halting the disease progression. Let's explore how biofungicides are already making a difference in the field. First is Trichoderma spp. This remarkable biofungicide is a powerhouse against devastating fungi like Fusarium and Ganoderma, two pathogens that cause massive crop losses worldwide. Farmers use it not only for protection but also for improving soil health as it enhances microbial diversity. Second is Lestani, a patented innovation. It is a biofungicide specially designed to combat Ganoderma boninase, a major threat to oil palm plantations. It contains phenolic acids which not only limit fungal growth but also protects the plant's root structure. Think of it as a plant's underground armor.
Hello, my name is Muhammad Arif. So I will be presenting about the screening potential organism. So there are three criteria for selection, which is first the antifungal properties, second is compatibility with crops, and third is the environmental safety. First is the antifungal properties. The microorganism should exhibit strong antifungal activity against target plant pathogens, and it must demonstrate mechanisms such as competition, production of antifungal metabolites, and cell wall degrading enzymes. So second is the compatibility with crops. The organism must be non-toxic and non-pathogenic to the host plant and compatible with beneficial organisms in the rhizosphere. It should enhance plant health by promoting growth or providing resistance against stresses. And third is the environmental safety. The organism must be safe for the environment, posing no risk to non-target organisms, humans or animals. It should ideally thrive under organic farming conditions without harmful residue. There are two methods for screening potential organisms. First is in vitro screening, and second is molecular screening. I will give 3 examples for in vitro screening, which is first is the dual culture assay, second is agar-well diffusion assay, and third volatile organic compound assay. So for dual culture assay, pathogenic fungi and isolated microorganisms are co-cultured on solid media, for example PDA or nutrient agar. And then we measure the inhibition zone or the reduction in pathogen growth around the bioagent. Second is the agar-well diffusion assay. Crude extract or culture supernatant of the microorganism are placed in wells of agar plates pre-inoculated with the pathogen. The size of the clear zone or inhibition zone indicates antifungal activity. And lastly, volatile organic compound assay. We assess the ability of microorganisms to release antifungal VOCs in sealed petri dishes, inhibiting pathogen growth without direct contact. The objective of molecular screening is to confirm and enhance understanding of the bioagent's antifungal potential. So first, the example is DNA sequencing. Molecular identification ensures taxonomic classification and highlights similarities to known biocontrol agents. The second method is to use gene screening, which PCR-based method or genome analysis can identify gene encoding antifungal proteins, enzyme or metabolites such as TA for cetokinase production. This helps to pinpoint mechanism underlying antifungal activity.
Now I will be discussing about biofungicide formulation, focusing on its development, stabilization, and performance enhancement. Let's start with the formulation process. First, active ingredient development. The first step is screening and selecting effective biocontrol microbes such as Trichoderma species, Bacillus subtilis, and Pseudomonas fluorescens. These microorganisms play a key role on combating plant pathogens. Next is cultivation, where we optimize fermentation condition, adjusting nutrient, temperature, aeration, and pH to achieve high microbial yield. Finally, we process to harvesting and processing. The microbial culture are harvested via centrifugation and filtration and processed into wettable powder and granular or liquid suspension for easy application. So why this is important? Effective biofungicide formulation ensure first microbial viability is maintained to enhance plant disease control. Next is extended shelf life, which allow for long-term storage without losing effectiveness, and then improve application efficiency, ensuring ease of use for farmers. How to ensure biofungicide stability and shelf life? First, temperature control. Microbial viability is temperature sensitive. Storing biofungicide in cold condition extend their shelf life. Additionally, we use chiroprotectant such as glycerol and terphthalose to protect microbes from heat stress. Freeze drying, lyophilization is another effective method to maintain stability. And then pH optimization. The pH of the formulation must be carefully regulated, because we use a buffer like phosphate or citrate to maintain an optimal pH range that keeps the microbes viable. Next is carrier material. For dry formulation, we use a carrier like talc starch and kaolin clay to prevent desiccation and ensure uniform distribution. And next is encapsulation technology. Advanced stabilization technique include spray drying and micro encapsulation, which coat microbes in protective layer. Polymer coating such as alginate and chitosan enable slow release and protect microbe from UV radiation and environmental degradation by integrating this stabilization strategy. We ensure that the biofungicide remain effective from production to field application. To further boost biofungicide effectiveness, we need to incorporate special additives that enhance the microbial survivability and performance. such as protectants. Microbes are sensitive to environmental factors, especially UV light. So we need to use lignin, skim milk powder, and polyols as UV protectants and desiccation preventer. And next is spreader and stickers, which for biofungus apply via spraying. Uniform coverage is essential uh, to enhance the leaf adhesion and rain resistance, preventing wash off and increasing effectiveness. And lastly, nutrient supplement. Now, some formulation include nutrient booster to support microbial activity in the field, such as yeast extract, amino acid, and humic acid are commonly added to provide essential nutrients that help microbes thrive. Now, testing biofungicide in controlled condition. Before they can be introduced for large-scale agriculture use, biofungicide must undergo rigorous small-scale efficiency trial in greenhouse or growth chamber. So, small-scale efficacy trial. This control trial focus on several key aspects, such as fungal inhibition. We measure how effectively the biofungicide reduce pathogen growth, particularly against Fusarium, Phytophthora, and other diseases. And next, plant protection. The goal is to evaluate the biofungicide ability to prevent disease progression, ensuring plant remain healthy. Next is microbial viability. Since microbes are living organisms, it is crucial to confirm that the formulation maintains a viable microbial population over time. And lastly, application compatibility. The biofungicide must be tested for sprayer compatibility, ensuring it does not clog equipment or settle out in suspension. And next, a key metric for this assessment to determine the effectiveness of biofungicide. We use specific quantifiable metric such as a reduction in disease severity. By comparing treated versus untreated plants, we can assess how well the biofungicide protect crops from disease. Next is colony forming units. This measures the viable microbial count, ensuring the formulation meets the regulatory standard for efficiency. Lastly, is crop health and growth. Observing the improvement in plant vigor, yield, and resistance provide insight into the biofungicide long-term benefits. This is several challenges and solutions for biofungicide. Firstly is shelf life extension. One of the biggest challenges in the biofungicide formulation is maintaining microbial viability over time. Since biofungicide contain living organisms, they tend to degrade, losing effectiveness during storage. So the solution is advanced drying techniques such as spray drying, freeze drying, and can be controlled storage condition, including temperature and moisture regulation, prevent the microbial degradation. And next, B, uniformity in application. For biofungicide to be effective, they must be evenly distributed across the plant surface. However, spore-based formulation can clump together, reducing coverage. So the solution is using spreader and sticker. It promotes the addition and ensure that the biofungicide remain on the plant surface longer. And also can optimizing formulation for sprayer compatibility, prevent clogging. And next is field performance consistency. Because environmental factors such as humidity, UV exposure, and temperature fluctuations can negatively impact microbial activity in the field. So the solution is adding UV blocker and stabilizer protect the microbes from the degradation. Also can encapsulate technology allows for gradual microbial release, ensuring long-term effectiveness. Lastly, the regulatory and market acceptance. A major barrier to commercial biofungicide adoption is strict pesticide regulation that require extensive safety testing. So the solution is conducting toxicity and environmental impact assessment, ensure that the biofungicide meets the safety requirement.
Now I will talk about the 6 steps in methodology for field validation of biofungicides in organic farming. The first step will be the site selection step. The process begins with selecting diverse test sites that represent real-world organic farming conditions. We consider soil types, cropping systems, and climate variations to ensure the biofungicide is tested in environments where it will be applied. For example, Bacillus subtilis was tried across different soils to assess its adaptability and efficiency. The next step will be the experimental design. Next, we establish a robust framework using designs like the randomized complete block design. This includes replicating treatments and setting up control groups such as untreated plots with synthetic fungicides to compare the results. The next step will be the application of biofungicide. The biofungicide is applied using appropriate methods such as seed coating, soil drenching, or foliar spraying, depends on its formulations. Application, timing, and dosage are optimized for maximum effectiveness. The next step will be the data collection step. We then gather critical data including disease incident, severity, and plant health indicators such as growth and yield indicators. These measurable outcomes demonstrate the biofungicide's performance under real conditions. The next step will be the statistical analysis step. The data is then analyzed using statistical tools like ANOVA to identify significant difference between the treatments. This step validates the reliability of the findings and determines the most effective application conditions. The last step will be the evaluation and reporting steps. This step involves compiling trial data into regulatory compliance. That's all from me. Thank you.
So in conclusion, biofungicide offer an environmentally friendly and sustainably alternative to chemical fungicide by leveraging a natural microorganism to combat plant pathogens. Their effectiveness is driven by various mechanisms, including direct antagonism, competitive exclusion, and systemic resistant induction, while formulation and field validation ensure their stability and efficacy. Challenges such as shelf life, uniform application, and regulatory compliance must be addressed. With continued research and development, biofungicides have a great potential to enhance crop production while promoting eco-friendly agriculture practices. That's all from us.